So I am very much a scientist. It's a huge part of my personality. So I'm going to use this presentation today as kind of part of an experiment. Because what scientists do is they make observations, they'll form a hypothesis, they'll do an experiment, collect data, refine their hypothesis. And some time ago, I made an observation, and that is atheists and the skeptical community as a whole is always very supportive of science and scientists. And it's not only supportive, but they're also very interested in the topics. You talk about something like evolution, where there's individuals who spend their entire lives studying this topic, and you can go to an atheist skeptical convention, and you've got people like total laymen in the audience that have a very good, strong understanding of those topics, and they've just done it on their own. And I think that's absolutely fantastic. But then there's other aspects of science, of being pro-science, like being pro-vaccine. Most in the atheist and skeptical community is very pro-vaccine. They understand that it's a scientific consensus that these are very good things, and so we all go get our shots and make sure our kids are vaccinated. But if you take somebody, let's pretend there was a creationist standing up here, and they said, oh, evolution isn't true because if we evolved from monkeys, why are there still monkeys? I bet quite a few of you in the audience could refute that. Or how some creationists say that, oh, we can't have evolved because there's no way of generating new information in the genome. I think quite a few of you could talk about gene duplication and divergence and the different ways that we get new information. However, if there was an anti-vaccination person standing up here and they said, well, I don't want to get vaccines and I don't want to vaccinate my children and expose them to all those toxins, what's it matter to you whether I vaccinate my kids or not? And I'm not sure as many people would be as confident in their refutation of the anti-vaxxer as they would be of the creationist. And so as someone who describes themselves as an evolutionary biologist, I study the evolution of a retrovirus, HIV. And as someone who describes themselves as an immunologist, I don't think that the skeptical community needs to be as worried, as kind of intimidated by immunology and vaccines as they are, because the fact of the matter is, if you understand evolution, then you understand more immunology than you think you do. And you also understand some of the science behind vaccines, whether you know it or not. So what I would like to do today is just kind of draw connections between evolution and immunology and vaccines. So you all can go out into the wild and be as confident about defending vaccines as you are about creationism. Do I have to point it at something? Is there batteries in this? Maybe, yes, what is this? Here's wait a second, go forward one. Yay! So, like William said, I'm getting my PhD in immunology and microbiology. I'm in my 5th year right now. I've been studying this stuff forever. And I am going to try to cram a lot of information into about a 30-40 minute talk here. So just the very basics of immunology. There's some aspect of how you protect yourself from pathogens that is completely and utterly passive. Think of like your skin or tears or mucus. Really all that's doing is creating a barrier between you and whatever wants to hurt you. There is another component of your immune system that's a little more proactive about defending you, but it's not really smart. All it does is really recognize patterns. There are certain things found out in nature, in pathogens, that is never found in humans. So there are certain kinds of bacteria that have a special kind of sugar on their surface that humans never, ever, ever make. So you've got this little receptor that when it sees that weird sugar, it tells your immune system that something has gone wrong, this bacteria is around, you need to do something about it. And then in another case, So humans, we carry our genome as DNA. It's just really a storage mechanism. That gets turned into a message called RNA that turns into proteins. The proteins are what make up you and me right now. Some viruses don't have a DNA step. They carry their genome as RNA. So to make their messages, they have to have double-stranded RNA at some point. Humans never, ever, ever should have double-stranded RNA in their system. So you've got these receptors that when they see double-stranded RNA, they tell your immune system, be on the lookout, something's wrong. So these are just the very basic aspects of immunology. And the evolutionary history of just this kind of immunology, I mean, think about like plants and insects. They don't have the same kind of immune system as we do, but this is how they protect themselves from viruses and fungus and that sort of thing. But that's a different story for a different day. What I want to talk about right now is a different branch of your immune system called the adaptive immune system. And this is really where your all's knowledge of evolution kicks in, because it has everything an evolutionary biologist wants. Awesome. So your adaptive immune system actually changes in you over time in response to things you've been exposed to. So the adaptive immune system branch in you right now is not the one you had a year ago. It's not the same immune system you had 5 years ago, 10 years ago. It's evolved in response to the chickenpox you were exposed to, or dust mites, or the cedar pollen that has been driving me nuts this year. And it does this via random mutations. Evolutionary biologists, we love random mutations and natural selection and like making sure the most fit survive and the ones that are detrimental are killed. And a huge aspect of what goes on here is founded on gene duplication and divergence. That's a great way to introduce new information in genomes that creationists say can't happen. Yeah, okay. So I'm going to narrow things down again, and even though your adaptive immune system is a huge branch, I'm only going to focus on the B cells. So B cells create these things called antibodies. Some of you might be familiar with these guys, but it's okay if you're not. So what antibodies do, they are this Y-shaped structure. And the little arms of the Y, they recognize, let's say, a virus. And so if you've got a whole bunch of these antibodies sticking to a virus, that virus can't interact with the host cell that it wants to infect. So these antibodies have effectively prevented this virus from infecting you. These guys can also bind to bacteria. So those pattern recognition receptors I was telling you about, this lower stalk of the Y, there are some immune cells that if they see a bacteria with all these little antibody butts sticking off of it, they think, hey, that's a bad guy, I need to go eat him. They can also bind to bacteria, and if enough of them get together, certain bacteria, they can directly like blow these guys up all on their own. And then also, you know, cancer. Cancer is you, but it's an altered you. It's not supposed to be you. And so sometimes your immune system has a hard time fighting off cancers because it thinks it's just you. Well, sometimes cancers start making more proteins that they aren't supposed to, or just proteins in general they aren't supposed to make. And so antibodies can stick to cancers and tell your immune system, hey, I know you think this is supposed to be part of you, but it's not, and you need to eliminate it. So antibodies are a really super aspect of your immune system, and they function based on evolution. So the first thing I'm going to talk about is just super basic structure. An antibody structure is composed of two of these heavy chains, I've kind of highlighted in blue, and two of these light chains. So how do you make these? How does your immune system get started? Well, you start with a basic B cell that looks like any other cell in your body. It looks like a skin cell, liver cell, whatever, same genome. And what happens is, to make a heavy chain protein, you have to paste together different portions of your genome. This is not the case where you just make one big message, one big RNA that gets chopped together. The actual DNA gets cut into pieces and pasted back together. And so you have a V component, a D, an H, and a C component. And so these guys will ultimately come together, and then you get the RNA, and then you get the heavy chain. Well, the thing is, this isn't just one gene in your genome. Because of evolution, there are 51 different V's, 27 D's, 6 J's, and you've got 2 of this group as a whole because you get 1 chromosome from your mom and 1 chromosome from your dad. So when all of these guys are being cut and pasted together, you can get 15,000 different heavy chains alone as far as the diversity goes. But antibodies aren't just composed of the heavy chain, they're composed of a heavy and light. So when you go to the light chain, it's pretty much the same thing except smaller, light. So it's only a V and a K segment, and there's 40 different V's, 5 different K's, mom and dad. But then gene duplication and divergence, once again, this locus is actually duplicated, and you've got 31 different V's, 4 different J's, mom and dad. So just looking at the light segments, you can have about 100,000 different lights. So ultimately, when you look at each antibody composed of a heavy chain, 15,000 possibilities, with a light chain, 100,000 different possibilities that can go together in any particular permutation, you can have over 10 to the 8th different antibodies just based off of random mutation, evolution, basic stuff. But there's still more diversity going on here, because when those V and D and J segments go together, it's not snapping pieces of your DNA together like Lego blocks. DNA is not supposed to be cut. That's normally a sign that the cell is very damaged and it needs to commit suicide if you've got all these breaks in your DNA. I mean, it's cancer, melanomas, that sort of thing. It's a lot of breaks in DNA. But what happens is, you've got these DNA repair enzymes that see these breaks, and they don't want the cell to die, so they fix it. They're like, don't worry about it. It's like, I've got this under control. just move along. Everything's fine. The problem is, these repair enzymes don't really know what they're doing. They're just sort of BSing to prevent the cell from dying. And so what that means is, for every single VDJ combination, these junction regions could be absolutely anything that these repair enzymes think of. So the point is now that we've got absolutely a bazillion, gazillion different kind of antibodies in you right now, totally based off of evolution, randomization. So some of you all might be wondering, okay, antibodies are great. They bind to viruses, they bind to bacteria, they bind to cancers. We can get all this kind of diversity just like based off of germline. So why does anyone ever get sick? Because you would think, if you've got, like, we aren't even in real numbers anymore, like the bazillion, gazillion combination zone here. You would think by chance alone, you would have an antibody that recognized chickenpox, and yet everyone in this room has either had chickenpox or has had to get the vaccine to chickenpox to prevent them from getting chickenpox. So you might be kind of mad at your immune system, thinking, you're supposed to be so great, the creationists tell us you're intelligently designed, and yet we all still get chickenpox. What the heck is going on here? Well, don't get too mad, because the fact of the matter is, you do actually, oh wait, there's even more diversity, sorry. So I showed you that basic antibody before. There's not just that basic antibody. There are a whole bunch of different structures that, once again, due to gene duplication and divergence, they all have their own specialties. So lots of diversity is really the message I'm trying to get across here. So back to chickenpox. The fact of the matter is, just by chance alone, when you all were babies, you all had antibodies that could kind of sort of recognize chickenpox. So let's say, this is our chickenpox virus, it's purple. And our B cell is red and making our red antibodies. And my logic with creating this diagram is that red and blue make purple. So red kind of sort of recognizes purple here. The thing is, though, this kind of sort of recognition isn't really good enough to overtake the virus. It's not like a I don't know, a mob boss or, I don't know, a bouncer that sees this virus and is going to take care of the situation. This initial B cell and this initial antibody, it's more like a very snobby person at a party who like sees that you've worn flip-flops to their very fancy gala, and they're just across the room going, I can't believe this, I don't know. So they might go around and like talk about you, but they aren't gonna actually go up to you and be like, you need to put some fancy shoes on. But they do notice. These B cells do notice that chickenpox is there. And so what happens is a process called somatic hypermutation. So right now, you know, you've got skin cells that are constantly dividing and sloughing off and constantly dividing and sloughing off. Those skin cells right now are exactly the same as the skin cells you had when you were little. These shouldn't change at all. B cells are a little different. When they kind of sort of recognize something, they get these survival signals and these messages to duplicate themselves. And when they do that duplication process, by chance alone, they start creating these variations in the little arm regions of their antibodies, which means that the progeny B cells are just a little bit different from the mom and dad B cells. And so basic evolution, some of these B cells are going to make antibodies that bind better, some are going to make antibodies that bind the same, and some are going to make antibodies that bind worse. The ones that make better antibodies are going to be naturally selected, and then they're going to go on to divide once again. Natural selection, random mutation. Eventually what happens when you are exposed to a pathogen or when you get a vaccine is that you have these multiple rounds of mutation and natural selection when you get to the point where you don't have an antibody that kind of sort of recognizes chickenpox and is looking down its nose at chickenpox. What you ultimately get is a B cell that makes an antibody that really binds to that pathogen very, very specifically. So in this case, this final B cell, what you end up with is very much like a mob boss. When it sees this pathogen, it is on top of it. It's like, I want this guy dead, I want his wife's dead, I want his children dead, I want him out of here. And the thing is, it's like this mob boss sticks around, it remembers. So we've all had chickenpox. If you happen to babysit for a nephew or a niece that has chickenpox, you probably won't get sick again because you keep these B cells around. And what happens is they just sort of hang out. But then another immune cell will come up and say, hey, your homeboy's back in town. It's like, what were you gonna do about this? And this mob boss is on top of it to the point where you might be exposed to this pathogen again, but you never get sick. You don't even really know that it was there because this B cell took care of it before it became a problem. So that's fantastic in the sense of when you actually get sick, but you actually have to get sick. And as someone who had a very traumatic chickenpox experience, I'm very jealous of the kids these days that just have to get the shot. So the reason why vaccines work is that you can deliver things that look like chickenpox to train your immune system to recognize and to make these little mob bosses to chickenpox without actually having to get sick or any of the potential repercussions of that. You know, a lot of anti-vaxxers kind of glorify diseases and they think, oh, chickenpox is a rite of passage. It's like, and nobody dies from chickenpox. Well, that's not true. Before the chickenpox vaccines, even in the US, totally industrialized, people have access to fantastic medicine. You know, 120 kids used to die a year from complications due to chickenpox. Since that vaccine has been introduced, 3 kids a year. So the benefits of vaccination, the benefits of creating this antibody response artificially versus the actual infection is just completely unmeasurable at that point. It's so great. So there is a drawback to some of this randomness. I don't know, it's a drawback and it's a feature kind of, in that this is totally random. So if, for instance, 5-year-old you got the polio vaccine, you made an antibody response to polio. If we tested this evolutionary principle called contingency, you might have heard of this before when you think, if you go back and play the tree of life or play the tape of life over again, would we ultimately end up with humans? And probably not, because there are so many random events that occurred over our evolutionary history that we probably would have ended up with something very different than humans if we rewound the tape of life 4 billion years. And the exact same thing happens with immunity. If you give identical twins the exact same vaccine at the exact same time, they're going to make a different antibody response, just due to random chance alone. So what happens is, let's say we have these 8 individuals, and they are very responsible citizens who get vaccinated in accordance to CDC recommendations. And let's say, once again, that the goal here is a purple antibody, something that recognizes a purple virus. You can see via vaccination that quite a few of these people are on their way to purple. So the police lady, she's pretty purple, and the MD, she's purple, and the speaker and the painter are kind of purple. But due to chance, there are going to be some people who don't respond as well to the vaccine as others. And there's nothing you can do about it. You don't need to take any immune boosting supplements before you get a vaccine. Don't do that. This is random here. There are some people who just don't make that great of a response. And so the poor server over there, he's kind of in periwinkle land instead of purple. And the poor photographer, she's way off base with the blue. And the thing is, they aren't totally protected, but because everyone else is immunized, they're still protected. And that's especially important in the case of the poor carpenter there, who totally got his vaccine, and he just didn't make a good response at all. And that happens. I mean, if you look at any particular vaccine on the market, they might have a 95% infection efficiency. There's always going to be a small percentage of the population that just doesn't respond well, because we're infecting people with viruses that are extremely crippled, or dead viruses, or just components of viruses. Technically, you could put those into someone and they would just degrade without an immune response at all. And so sometimes, rarely, that happens. But these people are still protected. It's called herd immunity. So it's like in the case of if a wolf is hunting for deer. If there's just not enough deer around that wolf, it's going to ultimately die and go extinct. And that's what happens with pathogens. Things really start to break down when people quit getting immunized. So let's pretend these people never got their shots, whereas these 4 individuals still did. Sure, the MD lady is still protected, but the photographer isn't at all. She's still at risk of becoming infected because she doesn't have a protective antibody response. The poor waiter is still at risk, and of course the construction worker is still at risk. So what you need to take away from this is that I'm sure everyone in this room has gotten their MMR when they were babies. But there's those crazy anti-vaxxers who are not giving their kids the measles, mumps, rubella shot because they think it causes autism. And of course they take those kids on trips overseas, mission trips, and places where this disease is endemic. So these kids come back as these ticking time bombs, exposing everyone and anyone to measles. You might not have a protective response to measles, but you just don't know it because you're surrounded by people who are vaccinated. If that kid came in and sat in the middle of the room infecting all of us, there's going to be a percentage of us that still gets sick even though we've gotten the shot. So to kind of pull that into what I said at the beginning, if someone comes in and says, why do you care whether my kid is vaccinated? Why do you care whether I'm vaccinated? Well, you care because them getting vaccinated protects you too. It doesn't just protect the kids who can't get immunized. It doesn't just protect the elderly who maybe can't get immunized, the people on immunosuppressive drugs or chemotherapy. We always think about them. But anti-vaxxers directly affect you too, and you won't know it until you're sick. And there's nothing you can do about it at that point. So I've really talked about to this point about the awesome evolution and randomness in the immune system and what we can do. So really, why does anybody get sick anymore? Why don't we make vaccines to everything? Like, I don't want to get sick anymore. I don't want the flu ever. I got that once when I was younger. Well, the trouble is, it's not just us that is evolving. The pathogens are evolving. Some cases that's not as big of a deal because they don't mutate very much. So, I mean, you know, measles, if you got like real measles in 1961, you probably aren't going to get sick from one of the mission kids coming back with measles. And you know, if we got the vaccination when we were a child and made a proper immune response, we will be protected because measles doesn't change very much. Things start getting complicated with the very quickly evolving viruses. So especially in the case of, let's say, influenza, everybody knows that every single year they've got this new formulation of an influenza vaccine out. And the influenza that existed in the wild in 1991 is totally different than the one we've got today. So even if you got influenza or you got the vaccine, you know, 25 years ago, you still have to get the vaccine this fall. And there's just nothing your immune system can do about it except get sick or get the vaccine. Things get especially worse when things start going even faster in the case of HIV-1. So if you look at a particular individual and you look at the kind of sequence diversity in an HIV patient, first day of January, you go back the first day of February and that population in that one person is totally different. A month later, it's totally different. So, you know, your immune system with HIV is totally screwed because it has all this diversity. It has this ability to mutate and adapt to the viruses that you're being infected with. The problem is HIV evolves one step faster than your immune system can. So even with antiretrovirals, even with the kind of technology we have today with HIV, no one's immune system can catch up with HIV once it's actually infected someone. So of course the goal then is to create an HIV vaccine. I mean, stop these new infections before they happen. Don't make someone's immune system try to play catch up in a game that they're never going to win. Well, this might be in one individual patient, and this is the kind of diversity in HIV as a whole. I've got like 100 stupid little pictures I did up there. But in the real world, there are billions and billions and billions of different HIV variants. And the deal is with HIV, contrary to what Michael Behe might say about HIV evolution, it is this insane workhorse that, you know, you always think about these, you can't make too many mutations or the protein's not functional anymore. HIV doesn't really have that trouble. It can make a ton of mutations and it's just as functional as it was 20 days ago. So what happens is you might immunize someone against that little variant right there. If they're exposed to any other variant, they're still infected. So you're like, okay, fine, then I'll put that one in a vaccine, or I'll put that guy in a vaccine. It doesn't matter. It's like the way vaccines train your immune system is to respond to this one kind of virus. And that doesn't work with HIV because there are a gazillion different kinds of viruses. So in HIV vaccine world, like, we really need to go back and study the core of just basic viral evolution and how we can use that in vaccine design to get an edge on this virus. So, just a quick summary. You know, you don't need to be afraid of really jumping into immunology the same way some of you might have jumped into learning about evolution when creationists started annoying you. Because, I mean, it is, if you understand evolution, you understand how your adaptive immune system works. It's just on a teeny, teeny, tiny scale. So vaccines, they work because they train your immune system to fight a pathogen without you actually having to be exposed to that pathogen and getting sick. I mean, you know, every vaccine has its drawbacks. If you're actually infected with a pathogen, you go through lots and lots and lots of cycles of the natural selection of those B cells. So you ultimately make really awesome B cells. In the case of vaccines, you're only exposed for a short period of time, so you make pretty good B cells. That's why sometimes you have to get booster shots to be not just pretty good, but really good. I mean, of course there's drawbacks, but in my opinion, they're a heck of a lot better than getting sick. And so the bad news is, with some pathogens, I mean, they evolve too. And it doesn't matter how fast we make our influenza vaccines, influenza is going to change. And it's a bigger problem on a bigger scale with HIV. So, you know, if you all have any questions you think of later, new ideas, that sort of thing, absolutely email me at that email address, or just Google ERV. I'm one of the top 2 hits. One of them will get you my way. And I just wanted to put a picture in there of my puppy because he is sitting at home moping right now because I'm not there with him. And he is, I call him Darwin's pit bull. So there you have it. And you know, I think I do have a little time for questions, maybe. There's William. So I'd love to take some. And I think she's going to bring us a microphone. Awesome. Microphone.
Well, let's say that one of your parents got a vaccine. Does that affect you in any way?
Kind of. So, if I go way back, one of the specialties, there's a variant of antibody. Oops. There we go. So my totally non-MD advice is to women who are thinking about becoming pregnant, because you know that's always planned, is to make sure that you get all of your vaccines and all your booster shots right before you're thinking about getting pregnant. Because what happens is when you're born, you don't really have a good adaptive immune system yet. It's still developing in your thymus. So what happens is you get your B cells from mom via breast milk. So the M's up there, you see the little J chain, and the A's also have a J chain. That actually means those antibodies are secreted. So just you, for instance, you won't ever have breast milk, but in your tears, so maybe if you have influenza antibodies in your tears, it keeps you from getting infected if somebody sneezes in your face or like in your nose or that sort of thing. So kind of, sort of, but it's only babies right when they're born who are breastfeeding who benefit from their mother's immune system. But otherwise, I mean, as far as like the recombinations go with the antibodies, you're part mom and part dad. You're completely unique from mom and dad because you're only inheriting one of those alleles from each. So yeah.
So I got a mic now. Beth, literally 10 minutes before you started your talk, sent me a text message and posted on Facebook, and she says hi, by the way. Facebook is now being used to encourage people to start attending pox parties, and I was wondering if you could address that a little bit.
Well, I mean, that's silly. We've got a vaccine. And this is especially important, like even those of you who don't have kids or don't even want to have kids, the problem is with chickenpox is that when you get older, it can reactivate and it's totally different. It's a totally different disease called shingles. You do not want this. Like older people, you definitely need to get that vaccine because I've just heard horror stories about shingles, and you know, just all I can say to parents who think that getting chickenpox is better. I have scars personally from this. I had, you know, walking pneumonia, double pneumonia when I was 3, and I was still running around and having a good time. When I got chickenpox, I was out, and it's one of my first memories about how miserable I was. And if I have the option of not putting my child through this, absolutely I'm going to take that option. I don't know how much time we have, so just like beat us down if we don't.
I heard that there's a reemergence of smallpox in the world. Are the older people like me who have been vaccinated for smallpox, is that still going to take care of us? Or? It
better not be smallpox because we actually, like, that's theoretically could be used as a biological weapon. And so we have a few vials of smallpox at the CDC database that are not supposed to be taken out except for biowarfare research purposes, as like responding to them. But yes, if you've gotten the smallpox vaccine, it's like you should still have some memory to it. But again, how good that memory is and how good the response is still and the thing is, you can always test that. I mean, if you went to a laboratory and said, do I have smallpox antibodies? Like, they could test that. I'm not sure what they would charge you for it, but it can be done. If you, just for your own personal well-being, that would make you feel better, they could probably do that.
Thank you.
If the HIV virus is constantly evolving, changing with all these different mutations, how does it still function the same way? And why can't you go into like that base functionality and kill it there?
There's two things with that. One, how does it remain functional? The thing is, we call it plasticity. It has an extraordinary genomic plasticity, which means that you don't have to have a very specific amino acid at a very specific location for it to still work. And the thing is, it's like this, it's a retrovirus. Half of your genome is retroviral. These guys have been evolving for millions and millions and millions of years to be very good at what they do, and they are. And so HIV is just doing what it can do, what it can get away with evolutionarily. As to whether we can target the more conserved, like structural regions, the good thing about antibodies is that they do see structure. It's like if someone gave you a basketball, you could feel that it was a basketball. But if someone put a soccer ball there, you would still feel basketball. And if somebody put any other large ball figure there, you would think basketball. And that's what antibodies do. So if we can figure out a structure, no matter what the sequence is, but if the structure has to remain constant and we can block that structure, then theoretically that would block HIV. The problem is, even if we know what we want the immune system to target, because of that randomness, we can't force you to make that kind of antibody. The only way we can, like if we identify this super awesome neutralizing HIV antibody, there's no way for us to make you make that unless we do gene therapy. If we take B cells out of you and we genetically modify them and put them back into you, that would totally work. That would not work in rural Zambia or a rice farm in Southeast Asia, is the problem. Okay, one more. Right on that side. Walk over here like that affects the speakers.
You mentioned you had a run-in or two in the past with Michael Abbie. What exactly has he argued regarding endogenous retroviruses and what response?
He doesn't say much about endogenous retroviruses. There are other creationists who are just tripping over themselves trying to explain away endogenous retroviruses and what they mean for evolution. For those of you who are unaware, like I was telling that fellow earlier, half of your genome is leftover virus guts that have infected eggs and sperm over our evolutionary history. And the thing is, they're junk. People say there's no such thing as junk DNA. Yes, there is. It's junk. Because if these guys, the retroviruses, if they weren't junk, they'd cause cancer and AIDS, like all of the retroviruses. They're junk. It's a good thing. But what creationists don't like about them is that you can literally look at these retroviral insertions because it's random, and you can trace the tree of life backwards. I mean, it's like a connect the dots picture. And they really hate this because it's really obvious evolution or evidence for evolution. Michael Abbie is a different story because he doesn't necessarily deny common descent, but my run-ins with him involved HIV because he doesn't know anything about HIV, but he decided to write a book about HIV anyway. That's always a bad idea. It's a very bad idea because he was saying that, like I said, HIV is this evolution powerhouse. It can pretty much do whatever it wants, and it laughs at our immune system. And he was like, oh, you know, this virus is so fantastic, but it looks the same as it did, 100 years ago. Minor problem, it doesn't. So yeah, he was saying there was no new genes and no new functions, but that's not really true at all. There are new genes in HIV-1 that help it infect humans better. We got HIV from chimpanzees, so when it crossed over into humans, it had different evolutionary hurdles to get over, and it responded accordingly. Furthermore, really strong evidence against his particular claim is that there's this accessory gene that HIV doesn't really need, but it wants. And it turns out in Australia in the late 1980s, early 1990s, is a group of people who got blood from an HIV-infected individual, and that HIV lacked this accessory gene. And so these people were doing great back in the 90s, like before we had AZT, before we had any kind of antiretrovirals. Everyone else was dropping dead. These people were doing okay. So scientists were like, well, hey, maybe we could use this delta Neph HIV as a vaccine. Minor problem. These individuals, Neph, like just through mutations in HIV, it started building that gene back from scratch, like Terminator 2, like pulling the liquid stuff back together after you've blown it up. And those people ultimately had to go on antiretrovirals because this deleted gene built itself from scratch again, which really does, I think, demonstrate the power of evolution and what it can do. Okay.